So, Joakim, you are a researcher here at Lund University.
Yes.
And what is your background?
So I'm a molecular virologist by training. I did my PhD here in Lund. And then I did 2 postdoctoral researches, one in Belgium at the Katholieke Universiteit of Leuven, and then one in Oxford, the University of Oxford, before returning to Lund 2016 to establish my own research team.
And what is your research focused on right now?
So as a virologist, working with viruses obviously, but I'm most interested in trying to understand virus-host interactions and how those interactions lead to disease. So I'm trying to understand the different steps there, and then when we have identified those mechanisms, of course we want to go on and find potential treatments against the viruses that we're studying.
Are there any specific diseases you are focusing on?
Yeah, so my favorite virus, if you like, is HIV. But more recently I've also transferred to working a little bit more on viruses, respiratory viruses like corona, for instance, RSV, and also influenza viruses. And then I have also been involved in a few projects working on hepatitis and chikungunya viruses as well.
You say that you have been focused on HIV. Why has that been interesting for you to focus on that?
Yeah, I think this is actually something that I quite often tell my students when I meet them the first time, as a kind of introductory story. So it all started basically halfway through my undergraduate studies here in Lund, where we kind of had a course which wrapped everything together that we have read up to that part. And we had 1 week about HIV, and we were divided into student groups, and then we were tasked to design a new vaccine or a cure against HIV. And I got really, really hooked during that week, and I instantly felt that this is what I'm going to do. So after that I contacted my tutor, then I was recruited for a summer project, then a master's thesis project, and then all the way up to PhD project. So that's how it started for me.
Wow, so you were actually trying to develop a vaccine against HIV?
Yeah, we didn't succeed, unfortunately. But it was a really nice exercise, I think.
Mm. And when it comes to viruses in general, why do you think they are so interesting to study?
What really fascinates me, I think, is that if you look at the virus particle as an entity, they are not really complex. They're quite simple. They basically have the genetic material, the genome, which is then covered by a capsid, which protects the genome. And then you have surface proteins that helps the virus to infect the specific target cells. So that's basically it. That is the composition of a virus. Still, they are one of the major challenges that we have towards disease against humans in the world, global health. So I think that really triggers me, that how something that simplistic can still be so complicated and difficult to treat.
How come that they are so complicated to treat when they are so basic in a way?
Yeah. That's a very good question. One of the major characteristics of viruses is that they have an unparalleled capability of harbor new mutations and change. So they are the perfect example of classical Darwinism, survival of the fittest, if you like. So they constantly change, and in comparison with humans, for instance, or other more advanced species, they can, as I said, accommodate a large amount of mutations, which we cannot, because in our case, most mutations will be lethal.
Yeah, and it goes very rapid, this change as well.
Really, really rapid, because the turnover, the replication cycle is something that happens within days. So really, really fast, from one virus infects a cell, and then the cell bursts and new viruses comes out from the cell ready to infect new cells. So that happens within days, really, really fast.
Is that also something that fascinates you? This, like that you can really study evolution in a very short period of time?
Yeah, definitely. That's one of my favorite topics, I have to say. So one thing that I specialized when I did my first postdoctoral training in Belgium was on molecular epidemiology and virus evolution within hosts as well. And what is really interesting is that over a period of time, viruses kind of accumulates mutations because they change all the time. These mutations can be viewed upon as footprints or fingerprints, if you like, that you can actually collect or associate to other characteristics. For instance, geographical place where the individual lived that got infected. Also time, so when in time were that individual infected. And then if you collect a large set of samples and you sequence the viruses from these samples, you can build a pedigree or a phylogenetic tree based on the genetic sequence to see how related they are. And then you can actually backtrack and see where in time and where geographically most likely those viruses were, all the way down to the origin of the viruses.
What are the biggest challenges today when it comes to HIV?
It's probably that what we see more and more is that the different resistant variants. So we have a list of antiviral treatments, therapies that are available. But then what happens today is that you when someone comes in and are HIV infected, you put in a combination therapy, which means that you combine 2 or 3 antiretrovirals. And that is because if you only give them one, that is called monotherapy, the virus will very quickly evolve and become resistant towards that medicine, and then you cannot use that anymore. But if you give 2 or 3, the virus are not able to evolve in a way that it becomes resistant to all of them at the same time, normally. So therefore it works quite well. But what we see more and more now is that some of the first-line treatment regimens is working less and less efficiently because people get infected with already resistant viruses towards these first-line treatments. And that is a problem. And we definitely need to look into more treatment opportunities due to that, I think.
Who are the most affected by HIV today, globally?
So by far the worst affected region is sub-Saharan Africa. So that's about 70% of all HIV infections are in that region. And with that comes also other challenges. So you have the numbers, many infected individuals, but then particularly maybe in some countries on the countryside, you may not have the capability to treat everybody and monitor people as well as you would like. So that is of course also a major challenge within the HIV pandemic, I think. So
last 2 years we have got this pandemic, COVID pandemic, but how can we prepare for the unknown viruses yet to come?
Yeah, that's also a very good question, also something that is being discussed. So as I'm sure you know, we do have influenza viruses that comes in new shapes every year. So every year there is a new influenza vaccine that you need to take in order to be prepared for that seasonal influenza. And since they have to start producing this vaccine prior to we actually know which influenza strain that will be the most common that year, they have to do predictions. And the way it works is that WHO is leading a large network of laboratories that over 100 countries are connected to or associated with. So those laboratories analyze influenza viruses in the nearby region, submit them to a common database that is then analyzed, and based on that projections can be done on which influenza strain are most likely to be the most common strain the following year. So then we can start produce the vaccine well in time. So that kind of work, I think, is something that we can really learn from also when it comes to other viruses, for instance coronaviruses. And we know that there are thousands of different coronaviruses circulating among bats, for instance. So if we start going to that source, characterizing those viruses, we can have a prepared strategy on how we can quickly design a vaccine against every one of them. And then we can just press the button when we see which virus is starting to blow up. Then we can do it quicker. So that is something that we can learn, and we can try to be more proactive in that sense. Another thing is, I mean, this was a respiratory virus now, the coronavirus. And that has been something that was discussed in Sweden, not at least, that having storages of mouth protection and other protective equipment, for instance, that are maybe governed by the government, so they can quickly be put in place when needed. That is something that we didn't have when this pandemic started. Mm.
So what impact can your research have in the long run, you think?
I think for me, what I'm trying to do in my lab is that we're trying to develop model systems where we can try to understand how viruses and human cells, human tissue are connecting and interacting with each other. The nice thing with these model systems, and that is something also that we need more research on actually in general, we have too few model systems to study virus in a really efficient way. And so what I'm trying to develop is model systems that can be used for a wide variety of viruses. Coronaviruses, RSV viruses, influenza viruses, HIV, hepatitis C perhaps. And if we can have those in place, and then together with those technology developments that I talked about before, to really produce large amount of data that can help us really disentangle those virus-host interactions and the mechanisms by which virus cause disease, I think that can really help us to discover new therapy and even new vaccines.
You work here at the Lund University, but of course you are also cooperating with other universities and other research groups around the world. How do you look upon the role with international cooperation when it comes to research and education?
Yeah, I think that's extremely important, really, really important. And if you go back and look at the kind of traditional scientists and looking at someone who is working alone, maybe on his attic or a chamber doing research, just thinking by him or herself, that is not working anymore, because the more we learn, the more we realize how complex things are. So we really need to have specific expertise, and we need to collaborate. So I'm trying to look at this as kind of a football team, for instance, where you have specific roles, specific expertise. You cannot win a football game if you have 11 goalkeepers or 11 strikers. You need to have people with different expertise. And that is the same in science, and that becomes increasingly important, also with the technology developments that we see. So that's also why, both nationally, locally, nationally and internationally, we need to create a critical mass of researchers that have common interests, but also different interests that can interact together and then, as a team, move boundaries.
Joakim, thank you for this interesting coffee break.
Thank you so much. It was a pleasure.